Welcome student in the next lecture of our first week, and in this we will be talking about plant cell wall. As I have told you that we will be discussing in somewhat more detail about plant cell wall structure because it is a very important component of anatomical feature of a plant. Now, when we talk about cell wall, it is an outer rigid covering of plant. Every plant cell will have a cell wall. Suppose this is a plant cell. So, it will have a cell wall, and inside this plant cell wall, plasma membrane will be present, and inside it cytoplasm with various organelles will be present. Now, cell wall is basically transparent or almost translucent in nature because it does not contain chloroplast or any other colored pigment. The main purpose of plant cell wall is to provide protection, to maintain shape of the plant cell, and to account for plant turgidity. Now, the plant needs to be turgid. Turgidity is actually when the plants take up water, it swells and it becomes turgid, and that swollen condition is known as it becomes turgid. Now, when the cell becomes turgid, the walls of the membrane exert pressure on the outer side, it exert pressure on the cell wall, and if this cell wall is not rigid or if it is not present, the cell can burst. So, this cell wall protects the cell from bursting when it is in turgid condition. Now, turgidity is also required because in plants, cell actually grow by absorbing water. By the movement of water, it performs various functions like cell elongation. So, when cell absorbs water, it elongates and it helps in the growth of the cell. It also helps in conduction of various minerals and various food particles to different parts of the cell. So, the absorption of water and this turgidity is important to perform various functions, and this cell wall helps in performing all those functions and maintaining the shape of the cell. It also helps in protection because it prevents entry of any foreign particle into the cell. Now, plant cell wall is basically made up of a polysaccharide which is known as cellulose. Now, cellulose is a polymer of beta D-glucose. So, lot of these monosaccharide, this D-glucose sugar units are joined together to form a very long polymer which is polysaccharide. Now, these polymers, these long thread-like polymers or these cellulose molecule, they combine together to form a structure known as microfibrils. Cellulose is one of the most abundant carbohydrate which is found on earth, and it is commercially very important. Cellulose is extracted and it is used in making lot of commercial products like clothes and paper. Now, basically cellulose is hydrophilic in nature because it contains lot of hydroxyl group, but because of its large molecular structure and because of binding of so many microfibrils together, it is insoluble in water. Now, in this different layers of cell wall, cellulose are embedded and these microfibrils are oriented in various ways to contribute different structural organization of the cell wall, that we will discuss here, that how this whole thing is possible. Now, this is the structure of beta D-glucose, which is the monomeric unit of cellulose. Now, beta D-glucose means that in this glucose molecule, the OH group which is there on the first carbon is above the plane of this molecule. So, this is D-glucose, this is beta D-glucose, otherwise it will be alpha D-glucose. If this OH group is on the lower side of the plane, it will be alpha D-glucose. Now, these glucose monomers, they join together by beta 1,4 glycosidic bond, and now lot of these glucose molecules join together, about thousands of glucose monomers join together by beta 1,4 glycosidic bond, and they give rise to a cellulose molecule. Now, when they form this beta 1,4 glycosidic bond, we can see here that the adjacent glucose molecule is inverted in orientation. Now, you can see there that CH2OH group is on one side of one glucose, however, it is on different side on the other glucose molecule. So, this orientation is applied to form a straight chain molecule, and in this way a long chain of cellulose molecule is formed. Now, many such long chains of cellulose molecule, they attach themselves now lot of hydrogen bond formation takes place between these cellulose molecules, and they attach together to form a structure which is known as microfibrils. Sorry. So, they form thread-like structures in which lot of cellulose molecules are attached together. Now, this forms now lot of microfibrils, they also attach together to form a thicker rope-like structure which are known as macrofibrils. Okay. Now, these macrofibrils could be oriented in different ways in cellulose cell wall, in the wall structure. We can see here that lot of microfibrils, they can be oriented in various orientation, okay. And this type of orientation could vary in different types of cell wall. It could vary depending on the different species also. Even in the same plant also, different cells can have different pattern of this orientation of this fibrils, and this happens because these fibrils can polymerize and depolymerize in different ways and can be arranged in different orientation. Now, basically this cell wall contains 3 layers: middle lamella, primary wall and secondary wall. Now, coming to middle lamella, when we talk about middle lamella, now suppose it is one cell and adjacent to it there are several other cells also. So, a middle lamella will be found in between 2 adjacent cells. So, we can say that it is a common layer which is present between 2 cells. So, it acts as a substance which attaches or glues together the adjacent cells present in a plant. Now, basically we can say that it is the outermost layer and it is sticky in nature. It is very thin and it is not very easy to identify the middle lamella when we observe it under microscope. The composition of middle lamella is, it is made up of pectin, and some calcium ions are also there. Now, pectin is also a polysaccharide which is heterogeneous in nature. Heterogeneous means that it contains different groups of monomeric sugar units. Like in cellulose, we have only glucose molecules which were attached together. So, it was homogeneous in nature, whereas in pectin, many different groups of sugars are attached. Okay, different groups of sugars could be attached. Okay, and it is also branched. So, lot of branching can come in pectin, which is also made up of sugar unit. The most important sugar which is present in pectin is galacturonic acid, although many other types are also present. However, the composition of this pectin can change in different plant species as well as in the same plant also, in different organs also. The composition of pectin can change. Now, pectin could be dissolved by strong acid. If we want to isolate that pectin in lab, we can dissolve it with the help of acid. This pectin has importance during, we can observe this type of phenomena during leaf abscission. So, you would have observed that during leaf fall, the point where leaf is attached with the stem becomes very delicate, and even with a slight touch you can observe that the leaf can fall. So, at that point the joining portion where the two cells are attached together, at that point middle lamella is degraded with the help of enzymes. Now, because of that degradation, the leaf is more easy to fall. So, this is known as leaf abscission. During fruit softening also, the middle lamella gets dissolved, and that is why we can observe that the fruits are more soft when they are more ripe. Because of this property, pectin is also used commercially in making jams and jellies. Because they have gluing nature, they can attach the substances and form a gel-like thing. So, they are used in making these things. Coming to primary wall, now every cell will have a primary wall. Now, primary wall will be adjacent to middle lamella. Suppose this is a common wall, suppose these are the two adjoining cells. So, along every side of each side of this middle lamella, there will be a primary cell wall. So, if this is the middle lamella, this will be the primary cell wall. Now, you can see here that on either side of the middle lamella, this primary cell wall is there. Now, these are attached also together with the help of plasmodesmata that we have already discussed before. The primary cell wall is basically thin and it is flexible, it is extensible. This primary cell wall is actually found in all plant cells, but basically it is more evident in dividing and growing cell because it is flexible in nature, and when the cell is dividing and it is elongating, it is required that the cell wall do not resist that growth. So, for that nature only primary cell wall is present in dividing and growing cell. This primary cell wall, it contains cellulose fibers, that is natural, every cell wall will contain cellulose fibers, but along with that it has many other components in the matrix, which you can say the viscous thing in which the cellulose microfibrils are present. That matrix, hemicellulose and pectins are present, the two most important components along with cellulose. Hemicellulose, you can see here that it is basically a cellulose, it contains sugar molecules, glucose molecules, but it is heteropolymer. That is, it is heterogeneous in nature. Along with glucose, it contains many other sugar molecules also. It is also branched and the size is slightly shorter as we compared with cellulose. Now, we can see here that along with glucose, it contains the 5 carbon compounds, xylulose, arabinose, 6 carbon compounds, glucose is there, then mannose, galactose, ramenose can also be present in hemicellulose. Now, the function of hemicellulose is that it helps in binding the microfibrils. See, when the microfibrils are attached or they are crisscrossed in different orientation, this hemicellulose molecules help in binding those microfibrils to form certain orientation. So, it also helps in regulating the cell wall expansion, because during cell wall expansion different orientation of these microfibrils occur. So, it helps in that process. Next important thing is pectin, which we have already discussed that it is found in middle lamella. It is heterogeneous in nature. It is also a polysaccharide, and the most important polysaccharide is galacturonic acid. The pectin composition definitely in the primary cell wall will also vary among different plant species and even among different parts of the plant. Now, along with the hemicellulose and pectin, a small amount of proteins are also present in the matrix of the cell wall. okay, and various enzymes are there. Now enzymes serves various function. Now many different types of enzymes are there. Many cell wall degrading enzymes are also there, like whenever it is required, the cell wall can degrade the cellulose component. When the pathogen attacks a cell, then to protect from the pathogen, many enzymes are synthesized which can degrade component of a pathogen. Similarly, cross-linking enzymes, the enzymes which are used in cross-linking the different microfibrils, they are also present in that matrix. Along with that, some lipids are also there. Now, you would have heard of cutins or the layer of cuticle on the surface of the plant cell. Waxes are also present, particularly in those plants which are there in desert environment. A thick layer of cuticle is there which prevents water loss. A thick layer of waxes are also there. Now, this cutin, they are basically non-polar compounds. These are all non-polar compounds. Waxes, they are all. So, they prevent water loss. Suberins, they are also lipophilic compounds which are present in the cork cells. They all help the plants to prevent the plants from drying. Now, this is just a structure of plant cell which is showing the plasma membrane on its inner side and middle lamella on the outer side, and in between you have a primary cell wall. You can see here a lot of microfibrils are there which are oriented in different directions, and pectin and hemicellulose are binding with these microfibrils, and they are helping in the binding of these different cellulose in different orientation. Now, coming to the secondary cell wall. Okay. So, coming to secondary cell wall. Now, secondary cell wall is not found in a dividing cell or in a growing cell. It is found when the cell has completed its growth period, it is not enlarging and it has completely stopped its growth. So, in that, because the flexibility after formation of secondary cell wall, the flexibility will be lost. This secondary cell wall, as we can see here, that it is a very thick cell wall, okay. Primary cell wall as well as middle lamella, which we talked about, were very thin, but as compared to that, secondary cell wall is very thick. It is made up of different layers. We can designate 3 layers like S1, S2 and S3 to secondary cell wall, but it's not necessary that all the 3 layers will be present in every plant species or in every cell type. Depending on the requirement, the composition of the layer as well as the presence of these layers can vary. Now, these layers can be different in their composition. However, cellulose will definitely be present, hemicellulose will also be there, then pectins will also be there, but their composition can change. Even the orientation of the microfibrils in different layers can change. So, all these changes can be present in all these different 3 layers. Now, along with that, a very important component of secondary cell wall is present of lignin. Now, lignin is an organic polymer. See, it's not a polysaccharide, it's a polyphenolic compound. Now, phenolic compound, you know that they are basically aromatic compounds, okay, containing an OH group, but along with that they would have multiple linkage of all these basic units, and along with that they will have many other compounds attached with it. So, the composition of lignin is also heterogeneous in nature and it can change depending on the cell type. But lignin is found deposited in all the secondary wall layers. Now, once the lignin is deposited, the cell will lose its elasticity, it will become rigid, it will lose its flexibility, there will be an increase in hardness, the tensile compression will increase, that is, it will not be able to break up in high pressure, and the permeability of water molecules will also be lost. Now, when the thickness of the wall will increase, we can see here that as the wall is increasing in thickness, the lumen, the cytoplasm inside the cell is becoming narrower and narrower, and because of this the protoplasmic content, or you can say the living component, will decrease and eventually they will die, because they do not have any flexibility of movement, water permeability is also lost. So, finally the cell will die. So, you can see here that presence of secondary cell wall is present in dead cells. The purpose is to provide mechanical support. First of all, when the cell is increasing in size, the cell wall is increasing its thickness, it is becoming more harder, so it will provide mechanical support. It is present in trachees, vessel, cork cells, sclerids and fibers, that we will learn later, that these all tissue provide mechanical support and also help in water conduction. Now, one very important component of secondary cell wall is present of different patterns of deposition of that secondary cell wall material on a plant cell. Now, when secondary cell wall is deposited, it's not that at every place it is deposited. Now, suppose we take an example of a tracheid or a vessel. These are actually elongated cylindrical cells which are dead in nature and have secondary cell wall thickening. It is observed that the secondary cell wall will be deposited in different fashion, like if it is deposited in ring like fashion, it is known as annular. It could be deposited in spiral fashion. Okay. We can see here that only cell wall thickening is present at those places, whereas rest of the thing does not have cell wall. So, okay, at these two types of thickenings, some sort of elongation or some sort of growth is possible, because it has many areas where secondary cell wall thickening is not present. Then we have scalariform, which is ladder like deposition. It is slightly more complex and more cell wall deposition is seen in this type of configuration. Then we have reticulate, which is basically net like deposition. So, in this case also the deposition is quite heavy and it is not found in growing and dividing cells, then you will have a deposition in the form of pits. Holes are there created in secondary cell wall. Then you will have simple pits. That means the entire cell wall has secondary thickening and leaving small areas where holes are created, means that at those points the lignin deposition is not present. Another case is there where you have pit but they are known as bordered pits. Now to explain this pit, suppose you have two cells are adjacent to each other, okay. When secondary cell wall deposition takes place, we can see that the primary cell wall as well as middle lamella is confined to a very thin line, and the primary cell wall is basically permeable because it has plasmodesmatal connections, and along with that you have heavy deposition of secondary cell wall. Now when we observe a pit, so basically this is a pit where the cell wall deposition is absent, creating a hole-like structure on either side of a membrane, and it is always observed that a pit pairs are present, means pit on one side is always attached with pit on the other side. So these pit pairs are there which are separated by a boundary made up of middle lamella and primary cell wall. And this is the pit membrane. Actually we can say that it's the pit membrane. So this is simple pit. When we talk about bordered pit, what we observe that suppose this hole-like structure is there, this secondary cell wall forms an arc-like structure on that hole. You will have a pit hole and the aperture, which is the aperture, and the chamber of this pit will also have a diameter. So you can see that the pit holes are smaller but inside the pit chamber has a wider diameter. So this is bordered pit, and many a times the pit membrane gets swollen in the center, and this is known as torus. Sometimes these pits are present only on one side, which is known as half bordered pit. It can happen if suppose this dead cell or suppose a trachea is aligned with a parenchymatous cell which is living, so only on one side the pit will be present. So this is known as half bordered pit. This torus can also help in regulating the flow of material between the pits. Many a times it has to block the entry. It can go and come and lie on this whole side, blocking the entry of the material inside the pit and finally inside the adjacent cell. So these type of modifications we can see in secondary cell wall, which will help in defining the function of a plant, because the presence of secondary cell wall as well as different formation helps in different functions of the plant that we will see when we will study different plant types. So this is all about for today. In the next lecture we will deal with another topic related to the first module. Now in the last, I just want to think about a question that why plant cells have a cell wall while animal cells do not have, means what are the important characteristic feature that makes a plant to have a cell wall while animals do not have it. So this is for today. Thank you.